Let me ask first, are there questions from any of the previous sessions? I see one question in the Slack tutors channel, and it is, can I add inhibitory Gaba synapse instead of Ampa in the point process manager? And the answer is yes. There's nothing particularly transmitter specific about anything the point process manager does with its built-in mechanisms. The alpha synapse can be given whatever time constant you want and whatever reversal potential you want. If you want it to be a fast conductance transient that has a depolarized reversal potential, then you will have something like an excitatory synapse. It's just a matter of using the desired parameters. If you want something that's slower and is shunting or is hyperpolarizing, you just set the time constant and the reversal potential accordingly. So if you want a hyperpolarizing, say GABA B type of synapse, you'll set the reversal potential to, say, something below the resting potential of your cells, something hyperpolarized. And if you just want a GABA A type of synapse, you can set its reversal potential to something close to resting potential. Okay. Are there other questions? Now, it is possible to have much more specific synaptic mechanisms than what you get with the alpha synapse or with neurons' other synaptic models, which I'll mention briefly. That's the expsyn and exp2syn. The ones we've talked about so far simply offer a time course parameters and reversal potential parameter. So the number of time constants, and you can set them to whatever you want. If you actually need something about the underlying molecular mechanisms themselves, you would need to implement that with something like NModel. In fact, the neuron model description language would be a good tool for doing that sort of thing. In this session, we're not going to talk about the use of NModel for implementing synaptic mechanisms or artificial spiking cells. We're going to talk about principally its use for adding new kinds of ion channels to cells. But you can do a lot with NModel. You can add lots of different kinds of equations for different purposes. So today we're going to focus on adding different ion channels. You can also include, you can add mechanisms for keeping track of ion accumulation. Diffusion can be represented with NModel. Transport mechanisms like active transport or ion exchange across membranes. It can handle anything, any process that you can describe with ordinary differential equations or kinetic schemes for reactions. And you can also include algebraic reactions, equations. That is, for example, you might want to implement a waveform generator like a swept sine wave. That can be done with NModel. Synaptic mechanisms, aside from alpha synapse, synaptic mechanisms typically use events. And today we're not going to talk about events, but you can add state machines, artificial spiking cells, and things that generate and respond to events so that you can implement highly sophisticated experimental protocols, for example, or construct complex stimulation schemes. All right. Well, why bother with NModel? Why don't we just use Hoke or Python? Well, the NModel has certain advantages. The principal advantage of NModel over Hoke or Python is that the statements written in NModel get translated into C and then into machine language instructions. And so you have high computational efficiency, orders of magnitude faster than you would if you tried to do something that required executing interpreted code like Hoke or Python. NModel is offers you a syntax for creating your new kinds of ion channels or synaptic mechanisms in which you only have to focus on the dynamics of your channel or your synapse. You don't have to be concerned about what kind of integration method is going to be used to generate the numerical simulation. And that's an important point because Neuron has several different integration methods, and they all expect different things from these mechanisms, and NModel will give it to you, will generate the necessary code to work with any of these. Another advantage of NModel is that it's very compact. One NModel statement is translated into many C statements, and as I said, the interface code is generated automatically. A big advantage is that you can you can implement mechanisms and models in which ionic currents are kept track of and influence ionic concentrations. And oftentimes in modeling electrical signaling in neurocells, we make the simplifying assumption that reversal potentials don't change, that ionic concentrations don't change. Well, what if you're operating on a problem where you need to keep track of the fact that concentrations can change? So, for example, you might be studying the effects of fluctuations of extracellular chloride or potassium concentration on cellular function. So you want any mechanism that generates a potassium current. In that case, you'd want Neuron to be able to keep track of that. Likewise with any kind of chloride current. And another important thing with these mechanisms is that NModel provides a way for you checking units consistency. And this is very important because oftentimes you have equations in which the parameters have different units. Concentrations might be not in millimoles per liter or moles per liter. They might be in molecules per square micron or something odd like that. NModel has units checking abilities that are very important, okay, to make sure that you get proper scaling of your numerical results. Now, we've talked about Hoke and we've talked about Python and we've talked about NModel, and the question is, how does all of this fit together? And this cartoon is meant to give an impression of how it fits together. At the core of everything is Neuron's computational engine, which is essentially a collection of data structures and compiled code. And what do you do when you're writing a model specification in Hoke or Python is you are writing statements in Hoke or Python that are going to modify existing data structures, make Neuron's computational engine create new data structures, and fill in parameters and variable values in those data structures, and call different functions and procedures that have been pre-compiled into Neuron. So that's what you're doing when you execute a Hoke statement or a Python statement. You're actually tweaking things in Neuron's computational engine. This arrow between Hoke and Python is to indicate that anything you do with Python, you can call Python from Hoke, you can call Hoke from Python. So if necessary in your code, you can execute a Hoke statement from Python or vice versa. Okay, but what if Neuron's computational engine doesn't have some particular thing that you need, some particular bag of equations, some particular function that you need to add? Let's say tomorrow somebody discovers a new ion channel with somewhat different dynamics than any previous ion channel has, or some new synaptic transmitter mechanism that involves a different kinetic scheme, a different set of state transitions. How can you add that to Neuron's computational engine so that then your Hoke or Python model specifications can make use of it? And that's where NModel comes in. When you're writing NModel code, you're basically adding new equations and potentially new data structures and new functions to Neuron's computational engine. Okay, as I mentioned earlier, they're all compiled and they execute at machine language speed. Now, if you look at NModel source code, it looks very different from anything you've probably seen before. What you see is a bunch of blocks, where each block has is marked by a keyword, then a pair of curly brackets, and everything in this curly bracket pair here belongs to the neuron block. And the neuron block is one of the blocks that's very important to the using NModel in the neuron simulation environment, because this is where you specify how Hoke and Python are going to be accessing whatever is in this model channel in this case, what parameters are available, what state variables will be available, and also how any ionic currents or ionic fluxes that this mechanism generates will affect Neuron's computational engine, and how the ionic concentrations and reversal potentials calculated by Neuron's computational engine will potentially feed back into this mechanism. So we see here a definition of a mechanism which is going to be, it says suffix here, so that means it's going to be a density mechanism, and the reason it says suffix is because in the Hoke programming language, the name of any of these variables that are exposed here, a G bar, for example, this G bar's name in Hoke is going to be Hoke underscore K-chan, and that underscore is reminiscent of the typewriter shorthand for a subscript or a suffix, and so that's the fully qualified name for this conductance parameter. Okay, would be G bar underscore K-chan. In Python, you can still call it G bar underscore K-chan, but you're probably better off calling it kchan.gbar, which makes certain aspects of Python easier to use with the mechanism. Okay, now there's other different kinds of blocks which simply define variable names, perhaps values of parameters, and units, abbreviations for units that are used in Neuron, in NModel. So in this NModel code, you would, if you have something that's going to be in units of millivolts, unless you want to be typing millivolt frequently in your source code, you might prefer to define MV in the units block as being equivalent to millivolt. Okay. Here's a parameter block that declares a variable a parameter called G bar and assigns it a value, okay, and says what its units are. And this extra little bit of syntax here is is used by neurons' graphical interface tools to specify the range over which this variable will be allowed to be set via the GUI. Okay, so it wouldn't make sense to have an ionic conductance with a negative conductance density. So the limits here are from 0 to 1e to the 9th, which is effectively infinite. Okay, there's a state block which defines the names of the state variables, and these are typically the variables that are going to be solved for in the course of running the simulation. So in the case of a voltage-gated ion channel, like a potassium channel, there will be an N state, a gating state called N, okay? And if there are intermediate variables that you need to calculate, or results of computations that you need to calculate and they're not parameters or states, those should be defined in the assigned block. And in this case, the whole point of this mechanism is to generate a potassium current, and that potassium current's name is going to be IK. It's declared down here as IK, and its units are declared as milliamps per square centimeter. So if we didn't know that suffix meant something that was a density mechanism distributed over the surface of the cell, by the time we get down here to read the parameter and see that that's in density units, that is in Siemens per square centimeter, that is conductance per unit area, and that it has an assigned variable, which is in terms of current density, which is milliamps per square centimeter, then we'd figure out that, yeah, this is a density mechanism. Okay. Now there's also a block for initializing states, and in this case, this initial block calls a function or procedure. It's a procedure which is declared elsewhere with a numerical argument equal to V, which is the membrane potential in the whatever segment this is inserted into, and then also assigns the value of ninf, which is going to be a parameter declared in the parameter block, to the state variable n. Okay, so that's what this particular statement would mean. Actually, you might not want to assign some fixed value to n. You might want it to be calculated by some procedure, and typically it would be calculated in a procedure. In this case, this one is called rates. It doesn't have to be called rates. It can be called something else if you like. But typically you would calculate initial values of state variables in a special procedure, or maybe right here even in the initial block with whatever algebraic equation it is. Okay. Now we're getting to the equation blocks. Okay, previously we were dealing with variables, okay, and interface to neuron. And the next blocks we're going to think about are the equation blocks. And these are, for example, the breakpoint block is very important because the breakpoint block is the block whose statements are executed at every new time step. So you advance from one time step to the next by executing the code in the breakpoint block, and Neuron's computational engine takes care of doing that for you. And here we see a solve statement where solve, deriv, method C, and exp tells us that, aha, this is going to be a block called deriv, and it's going to define a set of ordinary differential equations that are going to be solved by a method that's built into Neuron's computational engine called CNEXP. All right, and I won't go into any further detail there. That's spelled out in the Neuron book for those of you who are interested. And in addition, in the breakpoint block, that's where we also calculate the currents that might be generated. So in the case of a density mechanism like this, we're calculating the current that this mechanism generates as the product of the mechanism's channel density times its gating variables raised to whatever powers they're supposed to be. In this case, it's n raised to the 4th power, times the driving force, which is membrane potential minus the reversal potential of EK. And you'll remember in the previous slide, we had a statement here that said, in the neuron block, it says use ion K, which is the potassium ion, read EK, so it gets the reversal potential, I should say, it gets the equilibrium potential for potassium from neuron's computational engine, and write IK, which means it says this is a current that neuron's computational engine is supposed to include in the charge balance equations wherever this mechanism has been inserted, okay? So you're beginning to get an idea of how it is that when you say insert a density mechanism into a section, you'll see that it's going to, it may affect charge balance. Similarly, if you had a point process that actually used a potassium current and wrote, used potassium ion and read EK and wrote an IK, that would be added to the charge balance equation also for whatever segment the point process is inserted into. Okay. So we've considered now the breakpoint block, which is where we specify what equations are solved to advance from one time step to the next. And part of the breakpoint block here says you're going to be dealing with differential equations defined in the derivative block. Excuse me, the derivative block whose name is lowercase deriv. So here is, in fact, the derivative block lowercase deriv, and we see in here a call to this same procedure that was called during initialization, which basically updates the rate constants for our ordinary differential equation to be consonant with whatever the local membrane potential is, consistent with that local membrane potential. And the next thing we see in there is a first order differential equation. You'll notice the shorthand here uses a single quote to mark this variable as the derivative of n. And we see here a very familiar function to many of you. You'll see the time constant for the n state. You'll see the steady state value for n at this particular membrane potential, and you'll see the value of n. So what this is specifying is a first order lag between n and whatever the steady state value of n is supposed to be at a given potential. And the governing dynamic parameters are the steady state value and time constant, which are probably calculated in this rates block when we finally look at the procedure rates. And sure enough, there is a procedure rates, okay? And the procedure called rates expects a numerical argument whose value is local membrane potential in millivolts, and it contains algebraic equations that are going to calculate the values of n-inf and n-tau from the value of local membrane potential. Okay, so now we sort of walked through a skeletonized version of n model code for a potassium channel. Let's go to the next slide. So what do you do when you've got n model code that specifies a mechanism of some kind? Well, what you do is you open a terminal and you navigate into the directory where your n model code is located. And at the terminal, at the system prompt, you type the command nrniv model, nrniv modl, and that will launch a program that is going to compile all the mod files that it finds in that directory and generate a loadable library that contains all of the object code that was produced by translating those mod files into C and then compiling those mod files. Okay, and when Neuron then is started in that directory, it will automatically discover that library of compiled mechanisms, and it will fit those into its computational engine and make them immediately available to your GUI and to your HOK and Python programmatic interfaces. I include this little thing down here only for historical reference. There was a time when Windows users had to use this a little icon in the Neuron program group. They would double click on that, and then that would bring up a directory browser, and you would use that directory browser to navigate to the directory that contained your mod files. And when you got there, you would click a button that says choose this directory, and then what would happen is automatically those mod files would be compiled into a loadable library. But for it's much more convenient to just open a terminal and get into your working directory where your source code is. And when I say where your source code is, the easiest way to organize your code is for any given modeling project you're working on, make its own directory, put all the files that it needs in there. If it needs particular NModel files, if it needs particular HOK files or SES files or Python files, put them all in there. Change to that directory when you need to with a terminal, and when you need to recompile mod files because you changed one of them, let's say, or you add a new one, just run nrniv model. When you need to launch Neuron in there, if you're going to run Neuron, you're not working with Python, it's just nrngui and then space and then the name of whatever the main HOK file is. Or if you're using Python and you have a Python script that loads H and maybe GUI if necessary, and then loads maybe a session file or whatever HOK files are necessary or whatever Python code is needed. You can just do all that in that one directory, okay? Complicated directory trees are very elegant, and it's possible to create structures that prevent anybody else on the planet from using or reusing your code. So if that's your purpose, then that's fine. But keeping it simple is not a bad idea. Anyway, after you have compiled all your NModel code with nrniv model, if you then start the Neuron's GUI and you bring up any tool that uses mechanisms, you will find density mechanisms or point processes in their appropriate graphical menus. So here's this old tool which would bring up a single compartment model with a surface area of 100 square microns and present one with a set of checkboxes for all the available density mechanisms that could be inserted into it. And ordinarily, the only thing that you see in there is pass and HH, because that's what Neuron comes out of the box with. Those are the two density mechanisms that it has, pass and HH. But in this case, we see that two additional mechanisms have been added, one of them with a suffix HHK, the other one with a suffix KD. Okay, and we can select those by clicking on these checkboxes. And those checkboxes appear because when Neuron was launched, it was in a directory where the library that contained these compiled mechanisms was located. Okay, if you started Python and then loaded I should say import from Neuron import H, GUI, and then you clicked on the Neuron main menu's build and brought up the single compartment tool in that same directory, you would see exactly the same thing. You'd now have these two additional checkboxes. And likewise in the cell builder. The cell builder, you may remember, has a limited repertory of density mechanisms. And if you have compiled mod files in that directory and any of them are density mechanisms, they will automatically appear in the Cell Builder's biophysics menus the next time you open that channel builder, okay? All right. So let's talk a bit about what it is that distinguishes different kinds of mechanisms you can specify with NModel. We've been talking about density mechanisms almost exclusively, and these again are things that are, these are like ion channels that are spread over the surface of a section and or over the entire surface of the cell. And they're also called, as I say, well, they're density mechanisms. Sometimes you hear them called distributed mechanisms because they're distributed over the surface of the cell. So density mechanisms in the neuron block are characterized by a statement that says suffix and then the name of the mechanism. Then after that, if it's something that generates a current, it's going to have some kind of a statement about whatever the current is. In this case, the current is a non-specific current, which means this current will affect charge balance equations, but it will not affect the mass balance equations that neuron uses to keep track of ionic concentrations. Okay, and the reason for there being such a thing is that oftentimes people just want to assume that ion concentrations are constant, reversal potentials are constant, and that any transmembrane ion fluxes that might occur in the course of the experiment are not going to seriously affect ionic concentrations or reversal potentials. So if that's the kind of mechanisms you need, or maybe you don't even know what kind of currents are involved, you would declare them as non-specific currents. And you'll see that this mechanism has 3 range variables. Well, what are range variables? Well, a range variable is a variable that can have different values at different locations in space. We talked about range variables a couple of weeks ago. And so this is a density mechanism which can be inserted into a section, in which case every segment in that section will have it, and each segment's at a different place in space. So if this mechanism exists throughout a section, you certainly want each segment to be able to decide what its own leak current is, and it should be able to have its own local leak channel density. And maybe you even want there to be local variation of the reversal potential for the leak. So maybe you have this mechanism inserted in basilar sections and in apical sections, and you might say, well, we have experimental evidence that suggests the leak in the basilar region is a little more depolarized, and in the apical region it's a little bit more hyperpolarized. And so you want to have the ability to assign a different value to E depending on spatial location. So by declaring it range, that's what you get. Okay? Okay, so here's the parameter block that says what are the parameters here? One of them is the channel density, and we see it's in density units. So we know it's the channel density. And we also see that the reversal potential is fixed, it's constant. Okay, you can assign it any value you like in the course of a simulation, and it'll stay there until you change that. Okay, and by default, it will be minus 65 millivolts, just like by default, the G, this conductance density, will be 1/1000th of a Siemens per square centimeter. Okay, Mo is ohm spelled backwards. Before Siemens was adopted as the name of the unit of conductance. Okay, and what are the assigned variables? Well, V is assigned because V isn't calculated by this mechanism. It comes from neuron's main computational engine. So the value of V comes from neuron's computational engine, and automatically every mechanism can access V, can discover the value of local V by just including it in the assigned block, just declaring it, okay? And what about this I, this current which is generated? Well, again, it's calculated in the course of a simulation, so we need to declare it as an assigned variable, and it has units of milliamps per square centimeter. Now, does that make sense? Let's look at the equation that calculates it. Current is calculated as the product of conductance times driving force. Well, this conductance is in Siemens per square centimeter. The driving force is in millivolts. So a millivolt times Siemens is a milliamp. And of course, since it's Siemens per square centimeter, the product of G and V will be milliamps per square centimeter. So all of these units are consistent, and we will get numerically correct results by calculating this product. No need for any scale factors. Okay, let's look at the point process equivalent of this leak mechanism. In this case, we can think of something like a shunt. And what is it? Where does this shunt terminology come from? Well, when people were poking cells with sharp electrodes, they couldn't form a good seal between the sharp electrode and the membrane of the cell. So there would be like a little tear, a little rip in the cell membrane, and that was in parallel essentially with the electrode. So some of the current from the inside of the cell could leak out through that shunt. Okay, well, now one of the advantages of patch electrodes is that they form a gigohm seal around the mouth, between the mouth of the electrode and the membrane of the cell, and there shouldn't be any shunt. But if you have to represent a shunt, this would be a mechanism, a point process that you could use to implement a shunt mechanism. It's a point process, which means that it represents a current source or a voltage source, which is very small compared to a length constant for electrical signals. Okay, so in the case of a real rip next to a microelectrode, the tear could be half a micron or a couple microns or whatever, but that's effectively infinitely small compared to the size of any reasonable compartment. So it can be represented as a point source of current. Okay, and it is in fact, when you use this point process keyword, what you're getting is something which is defining a class, a new class of object. Okay, and one reason why this is important is because we might want to have many different shunts in our model. We may have many different sections with different segments, and we might want to have a shunt in each of those segments. So we would create a separate instance of the shunt class and attach it to every segment that we wanted to attach a shunt to or where we want to represent a shunt, the effect of a shunt. This also generates a non-specific current, not going to influence mass balance, but it will affect charge balance. And it has its own range variables. And we see that one of them is current, the current generated by the shunt. Another one is the reversal potential associated with the shunt. And this third one is R, which is the resistance of the shunt. Okay, so instead of talking about the shunt's conductance, we're going to talk about the shunt's resistance. And the parameter block declares the names of and the default values of the parameters. So in this case, the shunt resistance by default has a value of 1 gigohm, and its name is R. Its end model name is R. Okay. And it has a reversal potential of E in millivolts. Okay, and the default value of that is 0, because that would be the default reversal potential for a shunt that the outside surface of which is the bath. Okay, what about the assigned block? Well, we're going to be calculating this current that the shunt delivers as a product of driving force in millivolts and 1 over the resistance, okay? So it's going to be the driving force divided by resistance is going to equal the current that's generated. And Neuron's computational engine expects that point processes will generate currents in units of nanoamps, absolute current value. That is the total current produced by a particular shunt instance will be in units of nanoamps, okay? Not a density parameter, not a density current, but an absolute current, okay? And of course, V is from the main computational engine, and its values are in millivolts, its units are millivolts. So the breakpoint block now contains the equation that is evaluated to calculate the value of the shunt current at every new solution point in time. And we see here on the right-hand side, well, we see the driving force, which is membrane potential minus reversal potential. And we're not surprised at that. That makes sense. That's in millivolts. Divided by R, which is in megohms. So it's voltage divided by resistance, and the result is a current. But the units of this current are not going to be in nanoamps. It's going to be millivolts divided by, let me see if I'm getting this right here from mix. Yes, it's in millivolts per megohm. So why is there a 001 in front of there? Let me think this out.
It's in gigaohms.
Beg your pardon? Oh, R is in gigaohms. Yes, gigaohms, not megohms. Ta-da! So yes, and that would be picoamps. Okay. So a millivolt divided by a gigaohm, all right, would be in 10 to the minus 12th amps, picoamps. And we want a result in 10 to the minus 9th amps. Well, a picoamp is 1,000 times smaller than a nanoamp. So whatever this numerical value is, we have to multiply it by this scale factor of 1e to the minus 3. Okay, and it's in parentheses to indicate to us, the humans who read this code, and also to Neuron's model unit checking program which is called Model Unit, MODL unit, and MODL UNIT. And this tells Model Unit that this is a scale factor. Okay, these parens say that this is a scale factor. And Model Unit, if you left out this parens, yada yada, close parens, star, Model Unit would complain of inconsistent units and tell you what the scale factor had to be. And if you simply typed 0.001 times without the parentheses and then evaluated it with Model Unit, it would still complain because it wouldn't recognize this factor as being a scale factor. Okay, so you have to flag the Model Unit that it's a scale factor. That's what the parens are for. Okay. All right. And you can have many instances of this shunt mechanism attached to a given segment. Okay, so unlike a density mechanism where if you've inserted leak into a section, then every compartment in that section has got leak, and you can't insert leak a second time and get twice as much leak. You would have to change its channel density. With point processes, you can have many instances of them, and more than one instance can be attached to a given compartment. Okay. All right, so let's take a look at, let's compare these things along a different dimension. In this case, we've been looking at the end model code and comparing it for a density mechanism or a point process against each other. And in terms of their end model specification, let's see what they look like in the GUI. In the GUI, you would have to use the point process manager to insert or remove point processes from a section. Whereas for a density mechanism, you would have some other tool like a channel builder or this old single compartment tool, which would offer you a checkbox to insert or uninsert the mechanism. And if you've compiled these things, automatically the point process manager will have a shunt mechanism appear in its menu of point processes that you can insert. Likewise, if you had compiled the leak mechanism, all of the density mechanism handling tools will automatically get the additional user interface features, the additional widgets that they need to manage these new mechanisms, density mechanisms. Well, how do we manage them in code? Well, in Hoke, the statements would be of this form for a density mechanism. You have an insert statement, and then you'd specify the channel density for it, and you can print the current generated by it. And if it was a point process, you'd have to create an object reference, make that object reference point to a new instance of shunt attached to the midpoint of soma. It takes a lot of bouncing around to unpack this statement. The section where the shunt is going to be attached, leads, it's the first keyword on the line. The next keyword on the line is the name of the object reference that's going to point to it. Next, after the equal sign, is the new keyword, which causes creation of a new instance of the shunt class. Then we have the name of the shunt class, and in its parentheses, the range value at which the new shunt instance is attached. Okay? You get used to it after a while, but it's not very straightforward. And here we assign a value to the resistance. You notice we're using dot notation. The R belongs to the S, just as you would in Python. And then we can print the current generated by this instance of the shunt class. In Python, the statement that we would use to insert a density mechanism looks like this: the name of the section, the keyword insert, then the name of the mechanism. The full name of the mechanism is h.leak. Okay? And that's the name of the mechanism that, as from Python's perspective, is h.leak. And then we would be able to access the conductance parameter with this syntax, soma.leak.g, and then on the right-hand side, the numeric value that we want to assign to the parameter g. I mean, this is just standard Python sort of syntax. And then when we want to print it, we would say print, then the name of the segment, which is soma parens 0.5, close parens, and that would be the segment that contains the 0.5 location, dot name of the mechanism, dot name of the variable. Straightforward Python. Okay. If we want to insert, or I should say attach a shunt to the to the soma, we would say S, which is the new Python alias that's going to point to this instance of the shunt mechanism. S equals h.shunt, parens, then the name of the section, open parens, 0.5, close parens, which now makes this be the name of the segment. Okay, it says create a new shunt instance and attach it to the middle of the soma is what this is saying. Okay, and S is going to be the Python word that we use to refer to this shunt mechanism, this instance of the shunt mechanism. And then we can specify the value as the R parameter with this syntax, and we could print the current with, again, standard Python syntax. All right, let's see how this works out in a demonstration. Suppose we have Suppose we have a potassium current, a potassium ion channel specified by, I'm just showing you just enough of the N model code that you know it's a density mechanism. We haven't shown the name of the mechanism, but let's say we have a ion channel that generates a potassium current and tells neuron's main computational engine that yes, I'm generating a current and its name is IK and it is a potassium current. So keep track of it. And let's say that your section does not have an ion accumulation mechanism built in. Okay, sections don't come with an ion accumulation mechanism built in. They ignore the fact that that a potassium current is being generated as far as mass balance is concerned. So that if you just ran a simulation, you'd see that potassium extracellular concentration would remain constant, even though you're depolarizing the cell strongly and you are generating a lot of potassium current. Okay, so the experiment is the cell is starting out at rest, it's under voltage clamp, suddenly it's given a big depolarizing command and potassium current starts to increase. Well, extracellular potassium concentration is not going to change, it'll remain constant. And the reversal potential for the potassium ion is not going to change. It will remain constant. And consequently, the ionic current generated by our ion channel mechanism is going to go ahead and just increase and increase until it reaches whatever stable value is appropriate for the steady state potassium conductance at this new potential. Okay? So that's what these black traces show. If there's no ion accumulation, that's what these black traces are showing. But now if you include into your model cell, into this particular segment, or into the soma section, if you include a potassium accumulation mechanism, now let's take a look at this. It says it reads potassium current. So what it's reading is it's getting from neuron's computational engine the net transmembrane potassium current, okay? The net potassium membrane current. So in every segment, this ion accumulation mechanism will be able to discover what is the total potassium current density over my surface area, okay? And it calculates a new value of extracellular potassium concentration and writes it to neuron's main computational engine, which then uses that information to calculate new values for EK. How does the calculation work? Well, the calculation works through this breakpoint block, which tells neuron, okay, you're going to have to solve some ordinary differential equation. It's defined in a derivative block whose name is derivative state, and you're going to use this method, which is appropriate for first order differential equations. Okay, linear ordinary first order differential equations. Now let's take a look at the state block here, the derivative state block, and we see a first order differential equation. The left hand side says extracellular potassium concentration is equal to the potassium current divided by the thickness of the Frankenhauser-Hodgkin space divided by Faraday's constant. And what that does is this converts the potassium current into a rate of change of potassium concentration in the Frankenhauser-Hodgkin space, which is a region of limited diffusion in the cell, adjacent to the cell. And there is, so that's the thing that drives potassium concentration up. Now what's pulling it back toward the bath concentration is a first order exchange between the concentration in the Frankenheiser-Hodgkin space and the bath. So we're approximating diffusion of potassium away from the extracellular surface of the cell into the bath with a first order equation. And in fact, this works very well, and it fit the experiments that Frankenheiser and Hodgkin were doing. Oh gee, what was it, 50 years ago? Something like, maybe longer than that. Yeah, I think 60 years ago, in fact. So this is the equation that describes what's going to happen to the time course of the extracellular potassium concentration. So if we now do our experimental our simulation here, depolarize the cell, what's going to happen is potassium current is going to go up, and as it does, extracellular potassium concentration is going to go up. And as that happens, the reversal potential for potassium is going to become less negative. It's going to depolarize. Okay? And as that happens, the driving force for further potassium flux is going to change. We're going to find there's less potassium current. Okay, because our driving force for potassium to exit the cell becomes smaller and smaller. And our cell can't generate as high a potassium current. All right, so we'll get a more complicated time course of the simulation, and we'll get something much closer to what actually will happen in the real experimental case. So you can see how all this works out, and all you had to do was write these little bits here to get all this complexity, because Neuron is using the information that this mechanism generates potassium flux across the membrane, and this mechanism specifies what happens to that potassium that has exited the cell, and how does that affect the extracellular potassium concentration. Okay? All right. Well, you can accomplish other more complicated things with N-model, such as this implementation, this representation of events involved in transmitter release from a presynaptic terminal. In this case, we're dealing with a conceptual model in which there is a extracellular space and a presynaptic terminal whose membrane is right here. And all this stuff to the right-hand side of this line is stuff inside the presynaptic terminal. And what's going on is that when the cell is depolarized, calcium channels open up in the cell membrane, and a calcium current flows into the cell and becomes and adds to the intracellular free calcium. Once the calcium gets into the cell, it can do one of several things. It can diffuse into the core of the cell from layer to layer to layer. This is a cell that has a radial discretization of space, as if calcium is diffusing radially through shells, through circular shells that are concentric with the center of the cell. And so you have exchange between adjacent layers, and within any given layer, you have equilibration between the free calcium and a buffer. So that's what this transition is. So all these transitions are the buffering and unbuffering of calcium attaching to it a saturable calcium buffer. And we're saying it's the calcium that's free that can do the diffusing. We're saying the buffer is too large and too immobile to move between adjacent layers. But calcium can also do something else. The free calcium can bind with vesicles that contain neurotransmitter, in this case acetylcholine. And that when sufficient numbers of calcium ions have attached to a given vesicle, in this case, we're going to say 3 calcium ions, that transmitter is released. So there's a lot of transitions to describe here. There's this buffering and unbuffering. There's this radial diffusion. There's this binding between calcium and the transmitter pool that's ready to be released. And there's the release of acetylcholine and the breakdown of acetylcholine once it gets released. And then we're going to take for granted that somewhere else is a mechanism that generates a calcium current. Okay, so this transmitter mechanism can be implemented with a kinetic block in which there are several different state variables that represent the number of vesicles, the amount of acetylcholine that's been released, the activity of, I should say not the activity, but the presence of intermediate forms of acetylcholine bound to a cholinesterase. Okay, what is X? X is garbage, which is produced after acetylcholine is broken down, and then the concentrations of the buffer to which calcium has been bound, and calcium, which is free in each of the N layers of the cell. So we have all these different transitions. We have the binding between free calcium and the vesicles, which then releases acetylcholine. We have the binding between acetylcholine and acetylcholinesterase, which goes through a second reaction to then release garbage and freed acetylcholinesterase. And then we have the buffering reactions within any given layer, the transition between free buffer and free calcium to calcium bound to buffer and back again. And we have the diffusion between adjacent layers, okay? And the inward flux. And all of this, these equations describe this process, this conceptual model for transmitter release from a presynaptic terminal. Okay, and this was actually used in a published model that John Moore wrote quite a while ago. Okay, what about units checking? As I said before, NModel has a sort of a companion program called Model Unit, and if you have some NModel code and you want to check it for units, what you do is you type the model unit name, MODLUNIT, and then a space, and then the name of the mod file. Now, model unit will then open and examine that mod file and look for consistency of units. And if it finds that there's something that's inconsistent, it will issue an error message, and it will tell you what the proper scale factor is that you need to apply. They'll tell you what the error is, where the inconsistency is, and what is the scale factor you have to use. Okay, and I'm not going to take you through that today. We can do that some other time. But this is what the error message would look like in detail. And it says the previous primary expression with units of 1 minus 12 coulombs per second is missing a conversion factor and should read parens 0.001 close parens times and then brackets around it. So this expression needs in front of it a parens 0.001 close parens times to reconcile the inconsistent units, okay? And there is what you do with that kind of an error message, all right? So here's a little challenge that you can work on in your spare time. I'm not counting this as a homework assignment, but see if you can figure that one out. Discover what conversion factor will make this assignment statement consistent. We want to reconcile we want to apply a conversion factor to the right-hand side that makes the results of these products have units of micromolar per millisecond. Okay. Okay. So where do you find mod files? If you get Neuron source code from GitHub, and you get it onto your own machine, you look in a directory called neuron/source/nrnoc, and there it is. There will be stuff in there that you can use, you can look at. I'm going to bring over, in fact, such a file right now. Edit preferences. I'm going to make this a bit larger. Appearance. Let's make that bigger. Okay. So where are we looking? We're looking in NRN source NRNOC. So I'm in the NRN directory. I'm changing to the NRNOC, and if I look here, I see all kinds of mod files. Let me just do ls mod. Okay. And I think there's also some additional mod files elsewhere, but you can see the mod files of all of Neuron's built-in mechanisms, including HH. Here we are. It goes into some detail. Do we have an examples file? Okay. Great. Oh, well. Okay, so much for that. I'm going to go back to a more sensible terminal here. Okay. You'll also find mod files in ModelDB, and I put both of these URLs here. We're transitioning toward modeldb.science, but right now I think modeldb.yale.edu is more complete. But when you're getting mod files from other sources, be careful, because most user-written code is garbage. I said it. I should say an awful lot of user-written code is of relatively low quality. Sometimes the things that make it low quality is simply lack of organization and the presence of junk that doesn't do anything. Sometimes what makes it low quality is things that can actually cause bugs or frankly erroneous. So you got to watch out when you're borrowing code written by other people. There's certain sources that do exquisitely good end model code writing. You can probably guess who does that. Typically things that you'll get from the Allen Institute or the Blue Brain Project or from the core development group, Neuron's core development group, will be proper end model code. And there's a lot of people at EPFL who are doing very good programming in end model as well, and also developing some new stuff, which we'll talk about. If you want to find more mod files, you can also look in this reprint. I would get this article. It contains a lot of useful examples. It'll help you learn about end model. If you have the Neuron book or can get a hold of parts of it, chapters 9 and 10 of the book go into a significant amount of detail. about N-model itself and syntax and tips for writing N-model code. Sometimes people ask, well, why instead of borrowing stuff, why don't I just write my own? Well, the answer is it's kind of like, well, you might be able to, say, learn the ideograms for Korean so that you can sound out Korean words, but that doesn't mean you know Korean vocabulary and grammar. Or you might learn Cyrillic and can sound out some words, but it doesn't mean you know Russian. So just because you know a little bit, be careful. You should probably, if you want to do something that's special that nobody else has done already, try to find something that's close to what you want, and then make tiny changes to it and check the results to make sure you're getting what you want. Or forget about N-model and use the channel builder instead. And we're going to talk about the channel builder shortly. Where else to learn more about N-model? Again, that preprint that I talked about quite a while ago, but it's still, I should say, still quite relevant. There's a post on the Neuron forum that gets into discussing what integration methods should be used. That's in the solve statement. You remember it says solve and then the name of a block, method, and then we saw CNEXP. Well, if you want to know what methods to use for a particular kind of mechanism that you're thinking about writing or that you're looking at, read that post on the Neuron forum. There's some documentation about N-model in the programmer's reference, and you can read through these things as well, but they're not that, I mean, they're sort of good for reference at arm's length, but they don't give you a focused problem-centric example of things. So that you get much more of in the Neuron book or in this preprint. Okay, and finally I'm going to mention this development of this thing at the Blue Brain project. They have a GitHub repo, and what they're working on is something I think is going to be very important, especially as new processor architectures become much more widespread. Okay, so this is the homework I'd like you to do regarding N-model. And what you're going to be doing is a virtual knockout and rescue experiment. So what you're going to do is you're going to create a control model cell or a wild type model cell that has Hodgkin-Huxley ion channels, and you're going to verify that that it can generate a spike. Okay. Then after that, you'll knock out its potassium channels by reducing the HH mechanism's GK bar to 0, and you'll see what that does to its electrical activity. And then finally, you will rescue the cell's excitability by making it express a potassium channel that replaces the one that was built into the Hodgkin-Huxley mechanism. Okay, and the instructions here will help you get started, but you'll still have work to do. Okay. Okay. Now, as I mentioned the Hodgkin-Huxley mechanism, and we looked at HH mod briefly, I would advise very much that, let's see, let's look at that again. Not lost track of my own directories here. Neuron source, neuron OC. So let's look at hh.mod, and you'll see that it actually implements two different ion channels. the Hodgkin-Huxley, actually three, the Hodgkin-Huxley sodium current, the Hodgkin-Huxley potassium current, and the leak channel. And all that stuff is in one mod file. And if you're developing your own models, I would strongly advise against including more than one channel's definition in a mod file. Okay, so keep your code modular. And if you're writing a mod file to implement some kind of new ion channel, only include one channel in your mod file. It'll make it easier for you to debug and to reuse that mechanism with other models. Okay. All right. Let's see. I think I saw a couple questions. Are these blocks to be written in a specific order? I would follow the order that you see in the example references that I presented. The neuron block should come first. Unit blocks should come second if you have units. And then you start defining variables with the various variable blocks, and then you define equations. The order does not actually matter to the computer, but if you keep things organized, and there's an organized way to think about stuff, and there's a disorganized way to think, and I think it's much easier to think in an organized manner. If you follow a consistent pattern that makes sense, you'll get much farther, much faster, and you'll be able to debug your code much more quickly. So if you want a sort of an outline, the neuron, the Hodgkin-Huxley block, the Hodgkin-Huxley model is not a bad way to go. You could declare units first if you wanted to, and then your neuron block. But whatever you do, make sure the neuron block is right up at the top or near to the top of your mod file. Why? Because the neuron block is what you look at to discover quickly what are the names of things that are going to be accessible to Hoke or Python. So that's one of the things you want to know when you're writing Hoke or Python code. You know, what's the name of the channel densities? Oh, what do I call these things? You better look. You may have to look at the neuron block to discover that. Okay. Okay. So if you want to follow this pattern, that's fine. But just choose a pattern and stick with it. Okay. All right. If nseg is 1, then range variable is similar to a global variable, or is there some difference? No, actually, there's a difference. A range variable is not a global variable because a global variable applies to all instances of a mechanism. So if, let's say we have a synaptic mechanism that has a time constant and is a global variable. Okay, and we attach that synaptic mechanism to different compartments on a cell and to many different cells. If you change the time constant, you're changing the time constant for every instance of that synaptic mechanism. So if that synapse is attached to 100 different cells and you're using it, say, to represent an excitatory transmission in one cell, or maybe an inhibitory transmission in some other part of the same cell, and you change the time constant, if that time constant is a global time constant, you're affecting everything. Every time that mechanism is used anywhere. Okay. nseg is a spatial discretization variable. If you say nseg is 1, what you're saying is that the system is, quote, well stirred, or the system is, quote, compact, that spatial extent doesn't matter. Okay. And that's a very different thing than saying that something is global. Okay, because you can make models, model cells that for which spatial extent is nothing. So each one of them has an nseg of 1. But if you attach a, say you attach, say, a voltage-gated sodium channel to each of these things, and you want that sodium channel to have different densities in those different cells, you sure don't want that sodium channel density to be a global variable. It better be a range variable, or it's going to have the same value in every single cell. Is there a functional difference between soma insert HH leak and soma insert leak, or is it just alternate syntax? Yeah. Let's see, somebody looks like they've replied. It is a good question. Yes. Robert's answer is the right answer. Introspection. Okay. Okay, that's a good answer to look at in the Ask Tutors thread. Okay. All right. Okay, let's talk about the channel builder now. The channel builder can be used to create specifications of many different kinds of things, but it's limited to channels. You can't use it to implement pumps, buffers, diffusion, event-driven synaptic mechanisms, or artificial spiking cells. It's only for channels, and it can be used to, but it does a really good job of implementing voltage and or ligand-gated channels. And the gating in these implementations can use kinetic schemes or Hodgkin-Huxley style ordinary differential equations. And if you implement as a point process, if you're implementing a point process with the channel builder, you can actually have stochastic gating. And it is, in the past, it's been faster than equivalent end model mechanisms, but with the new optimizations that are being applied to end model in development, it may not be the case anymore. I'm not sure about that. Okay, there is a tutorial on the channel builder, and you'll find tutorials there. It wouldn't hurt for me to go to Neuron on the web here, programmers, no, we want to go to documentation. Guides and tutorials. Let's see. GUI tools, channel builder tutorials. And you'll see that this is in glorious 1990s style HTML. But don't let that put you off. It will take you, if you're able to do these particular exercises yourself without having to follow through the tutorial, you will have mastered the channel builder. So go ahead and work through this if you like on your own, especially the one with stochastic gating. It's a lot of fun. We're going to do just a tiny fraction of this today. So when you start working with the channel builder or any other graphical task tool, or for that matter, writing code, the first thing you need to do, though, is to think about what it is you're setting out to do. Conceptualize the task is the fancy phrase for that. And in the case of specifying the properties of a channel, the first thing that should come to mind is, Does it have ionic selectivity? I mean, that is a characteristic of a lot of real-world ion channels. They are ion selective. So what is that ionic selectivity, if it has it? What is the current voltage relationship? Is it ohmic, or is it described by the Goldman-Hodgkin-Katz equation? Are the dynamics specified with a set of ordinary differential equations, like in the Hodgkin-Huxley mechanism, or are they represented as a kinetic scheme or Markov state model? And what about the gates? Are there going to be independent identical subunits or different subunits? Are there going to be different subunits that have different fractional degrees of openness? That is to say, alternate gating states? What about sensitivity? Is the channel sensitive to, is it gated by voltage, or is it gated by the concentration of some ligand? And what about the transition style? In the case of Hodgkin-Huxley style dynamics, are you specifying the rates of transition in terms of alphas and betas that are forward and backward rate constants? Or are you specifying in terms of steady state and time constant that are functions of membrane potential or concentrations? And are these transitions governed, are these numerical values governed by functions, or are they governed by numerical values in a table, as it might be if you, say, are using experimental data and you haven't fit functions to the experimental data because maybe the data are too incomplete or too peculiar, and you don't know of a nice compact mathematical format for it. Okay, so those are the kinds of considerations that are going to come up in the course of using the channel builder. Here what we're going to do is we're going to implement the Hodgkin-Huxley sodium channel with the channel builder, and we're going to use it in a toy simulation where we're going to knock out the native Hodgkin-Huxley sodium channel, which is going to kill excitability, and then we're going to rescue excitability by inserting a Hodgkin-Huxley sodium channel that we built with the channel builder. Sort of like the knockout and rescue that I left you with as a homework assignment for the N model code. Okay. So this is the description, the mathematical description of our sodium channel we want to implement. Okay, so it's going to be alpha beta type of formalism. So what we're going to do is we're going to bring up a channel builder and use it to specify the channel's properties, basic properties, that is, its structures of, I should say, what states are important and what are its ionic selectivities, etc. What's it going to be called? And then we're going to specify the channel gating by the states, transitions, and how voltage and ligands affect the gating of channels. Okay, and that's our flow chart for what we're going to do. So first thing we'll do is we'll get a channel builder. And it's going to be Python 3 from neuron import HGUI. Okay. And I'm going to click here on build channel builder. And it will be a density mechanism. Okay. So there's what you get when you first create an instance of the channel builder class. And this thing comes up as configured as a leak, a density mechanism called leak, which has a non-specific ohmic ionic current, and the leak current is the product of the G leak parameter times the driving force. And the value of G is controlled by a particular parameter called Gmax. And the default Gmax is 0, and the default reversal potential is 0. So that's what we get when we first create a channel builder. Okay? So what do we need? We want to change its name, its ionic selectivity, its default conductance, and its equilibrium potential. Well, what do we want to call it? Okay, I'm going to click on Properties and select the name of the item to change. Well, we're going to change its name first. And you'll see that its name by default is leak. Well, that makes sense because it's that's what it started out as being. So we're going to change it to myNA. So here's the tool that came up when I clicked on Properties. Now I'm going to change this to myNA. Okay, so now we see the name of the density mechanism is myNA. Okay, what's the next thing we want to do? We want to say what its ion selectivity is. So I click on Properties, Selective for Ion. You'll notice how this menu from top to bottom is the sequence in which we want to use the tool. First thing we want to do is change the channel name. Now we're going to specify the ion selectivity. In this case, it's going to be specific for sodium channels. Other options are we could have it be a non-specific current. It could be specific for potassium, or we could create some other type of ion like calcium or chloride or who knows, lanthanum, whatever. Okay, so it'll be specific, it'll be selective for sodium channels. Okay, so now we see the channel description up here has changed to sodium ohmic ion current. INA is the product of G myNA times V times the driving force, which is V minus ENA. Okay. We want the default Gmax next, the default maximum conductance. Ohmic was fine, we didn't change that. We could have changed it Hodgkin-Huxley-Katz, but we don't, Goldman-Hodgkin-Katz, but we didn't want to do that. Okay, so let's get the default Gmax is going to be 0.12 Siemens per square centimeter. And there is the tool that pops up that allows us to specify a new default maximum conductance, 0.12 Siemens per square centimeter. You notice when we start out, we said it's a density mechanism, and so the Gmax automatically has units of Siemens per square centimeter, density conductance. Okay. What about its equilibrium potential? Well, automatically it's ENA. So, and this neuron knows about ENA, so we don't have to do anything special for that. We can now close this little panel here that we use to set the default Gmax if we want to. I can close that. I can always get it back if I like by clicking on Properties, Default Gmax. See that? There it is. So I can always throw it away because it's part of this. It's managed by this tool, part of the channel builder tool. Okay? So we want to construct gates. Properties, we've done our default Gmax. Oh, by the way, I could have cheated and just gotten a Hodgkin-Huxley sodium channel by coming down to here, and it would do the whole thing. But we're going to work through just to see what the user interface looks like. We want to construct gates, gate constructor. We could do that, or we could double click here. Select here to construct gates. So there we are. We have the gate constructor tool. And what we do is we create new states by dragging them from this palette over here onto the canvas. So we know that Hodgkin-Huxley has two state variables. One is M and the other one is H. So I'm going to click and drag the O over here. I'll click the C over here. Okay. because I know I'm going to need two of them. Okay. And so there's the states page. We just saw that it was empty. And it says click and drag O from palette, repeat for C, close. We've done that. So we have spawned the states. We're going to rename the states. So to rename the states, I click on the state I want to rename, and I don't drag it, just click it, and I get this tool up here that says change state name. I want it to change it to lowercase m. Accept. And the C, I want to change to lowercase h. There we go. So those are my two states. And when I've done that, I get the canvas looking like this. And now I want to go to the properties page. You'll notice I started out in the states page. That's our little radio button there. I can click on properties. And now what I want to do is I want to deal with the properties of M and the properties of H. So it says if I want to see the tool that allows me to specify the properties of M, I have to click on M. And I will do that in a second, but first I want to do a save session. File, save session. I'm going to call this draft.sesh. Okay. Now I want to specify something about M. And so looking here in this tool, I see up here, I see a power, I see an M fraction, and we remember that the conductance is the product of G bar NA times M cubed times H. So the power of M is 3, and the fractional conductance is 1. Don't worry about that. That's just 1 because when this is open, it's open. It accounts for all of the openness, okay? All right. Change the power to 3. And now I want to specify the voltage dependence of AM and BM. I want M to be governed by the alpha beta type equations. Okay, so we're dealing with M, and we're looking at M, and we're looking at AM and BM. That's our alpha M and beta M equations are exactly what we're dealing with. And we want to set the parameter values for them. Check the functional form for AM. It's not what I want. Here, the functional form that's shown is AM is just a constant. I don't want that. I want to have this one down here. Let me make that bigger so we can all see it. We want AM to have this form, A times X divided by 1 minus e to the minus X. Okay, where X is a K times V minus D. So AM alpha beta okay. Now how am I going to get the equations here? AM. A times X over 1 minus e to the minus X, where X equals K times V minus D. Okay, so I got that by clicking on equation type, AM, and that middle equation. This channel builder comes with equations that make sense for channel state gating. Okay. Next I want to, and I want to set the parameters. A is 1, K is 0.1, and D is 40. A is, have to be changed, 1, K is 0.1, and D minus 40. That's good. Now, for BM, we want to see which of these equations we want to use for B, for beta. And Bm is A times, it's just an exponential. Bm is A times an exponent raised to e raised to the k times v minus d. Okay? So equation type Bm, A times e to the k minus k times v minus d. Okay, we need to change these parameters, and those are going to be 4 and minus 1/18 and minus 65. That's 4, and this is 1/18, and that's minus 65. Whoops. Let me just make sure it's correct. 4 minus 1/18. Yeah, I thought so. Okay. Okay, so there's AM and BM, and we can look here and see these look like the typical textbook illustrations of AM and BM. And if we want, we can display the steady state and time constant form. U equals plot. Okay, so the steady state value for M approaches 1 as we make things more depolarized, and it's slowest right around the midway point, half activation point, and becomes faster the farther away you get from there, and that's good, that's what we want. All right, so now we want to take care of H. Okay, so we want to have H with a power of 1 and a fraction of 1. So click on H, power of 1, fractional conductance of 1. Okay, because remember, it's the product of M and H. Both have to be approaching 1 for the channel to be fully open. Okay, and now we're going to want to find the equation for it. So AH is the simple exponential form. Equation type for AH, simple exponential form, and definitely not those parameters. 0.07 minus 0.05 and minus 65. 0.07, 0.05, minus 65. Let me verify. Minus 0.05. And minus 65. So that takes care of AH. And BH is A over 1 plus an exponential function. Equation type, BH. Okay. Verify that. There we are. And the parameters for that are 1.1, oh, minus 0.1. And then minus 35. Okay. Which looks awfully like, oh, how about that? What a coincidence. Haven't messed up yet. Okay, file, save session, draft.sys, yes. Okay, 19, 20. Okay, well that's what we have to do to test this darn thing. we have now specified our channel's properties. Aha, that's what we want to do to test it. We want to build a cell builder that has this thing in it. Okay, so I have now specified all of this stuff in this tool, but this tool is a child of this channel builder manager. So I can get rid of this anytime I want. I can bring it back up by clicking on one of these equations. If I prefer to see the equation for M, okay, I can see the properties of M, and I can see the gating properties for H, okay? And I can go back and forth between them. Okay, so I can close this, and this thing exists now. I'm going to do file save session, and I'm going to call this nahh.sys. Okay, or let me call it myna.sys. Okay, so I can quit Neuron now. And if I restart with Python and I import H and GUI, I can do a H load file myna.sys, and I get back my channel builder. And if I look, build cell builder, and I want to make a single compartment model cell. So I get a single compartment just like that for free with the cell builder. I go to the geometry page. I want the soma to have a surface area of 100 square microns. Okay. Go to the biophysics page and look, now I have, in addition to RACM pass, extracellular, and Hodgkin-Huxley, I also have myNA. Let's put both Hodgkin-Huxley and myNA in there. So the cell is getting a double dose of voltage-gated sodium channels. Okay, let's come right over here, click the specify strategy box to now look at the parameter values for Hodgkin-Huxley and for myNA. I'm going to block expression of the myNA channels. So if I do that, I can do that in this model by reducing the conductance to 0. Okay, so now what I've got is a 100 micron diameter oocyte. It's a pretty tiny oocyte, and it is expressing the Hodgkin-Huxley sodium and potassium and leak channels, and it's not yet expressing the myNA channels. Do we have molecular biologists in the audience? If we do, please don't throw up on your shoes. Okay, all right, so now we're ready to actually do an experiment. Let's make an instance of this model cell. Actually, let me do a file save session. Okay, going to call it test rig sys, and it's got both the channel builder and the cell builder. Drag this cell builder down here. Now I'm going to hit continuous create, and I'm lucky. I don't have any error messages. Everything's working well. Pride goeth before a fall, right? All right, so now I have a model cell, I can work with it. I need a run control panel so I can run a simulation.
Ted?
Yes?
Could you just specify the geometry again? I mean,
the geometry of what?
The oocyte you have taken.
Oh, sure. There's only one section, and it has a surface area of 100 square microns, and it has an nseg of 1 by default. Okay, so it's a single compartment model cell with a surface area of 100 square microns. Okay, and go ahead. Question?
No, thanks.
Oh, okay. All right. So so there we are. We have our model cell. If we want to see a plot of membrane potential versus time, which graph voltage axis, and let me bring that graph over. And if I run a simulation, that's fine. There's nothing going on because there's no stimulus. If I want a stimulus, I need a point process manager. So it's tools, point processes, point manager. And I want a current clamp. Did I get an error message? No, I didn't. I didn't get an error message. Okay, how nice. So if I want to have a current clamp, I will select point process, come down to current clamp. It's going to be at the middle of the soma. I want it to be silent for the first millisecond. And then at 1 millisecond, I want it to generate a 0.1 nanoamp, excuse me, a 0.1 millisecond pulse of 0.1 nanoamp amplitude. That's easy to remember. And if I click on it and run, I get a spike. There's the ramp as the cell is being depolarized by the current that's being dumped into it by the current clamp. We get a little depolarizing ramp. Then the current being injected stops and the membrane potential starts to collapse towards zero, but the sodium channels are opening up and they continue to open up and they produce this nice spike. Let me make t-stop be 15 milliseconds. And let me just run that simulation again. So there we are. That's my spike produced by my control cell. It's the control spike produced by the normal cell, the wild type cell. It's not a wild type cell. It's an oocyte with sodium with Hodgkin-Huxley expressed in it. We'll call it the control experiment. And what I want to do is I want to make this stand out and easier to see. So I'm going to do color brush, make that be a thick black trace. Click there. Good. Okay. And then I'm going to close that, go back to crosshair. All right. Now, let me do the, you can think of the next step as either being pharmacological, like I just dumped tetrodotoxin in the bath, or as being, we do a knockout experiment and stop expressing sodium channels, and we wait a while, and the sodium channels all go away. So let's knock out the sodium channels. And the way we do that is by reducing GNA bar HH to 0. Now, if I do that, what is going to happen to the response of the cell to an injected current? Let me make sure I save this original response with keep lines. And the next thing I do is a new experiment here where I inject current into the cell, depolarizing current. Aha, it drives the cell from resting potential up a bit, but then membrane potential just decays away. It doesn't fire a spike. Okay? And it's not simply that I don't have enough depolarizing current, because if I make that current be many times larger, that's just a stimulus artifact. That's not a spike. I've just dumped a huge amount of current into the cell, and as soon as I stop injecting current, the current leaks out. I have opened potassium channels a bit, which is why we get this undershoot. Okay? And if you wanted to be sure about that, I could do a plot of current axis. Plot what? Soma. Okay. Ion A. Another one. IK. And I could do another plot what? Well, how about that? I produced by my NA. Okay, now let's run that simulation and let's do some color brush stuff. So potassium cools things down, so that's going to be blue. And Hodgkin-Huxley sodium current depolarizes the cell. I'm going to let that be a black trace. And the current produced by the MyNA channel is going to be red. Go back to crosshair, and let me do view equals plot. And the only thing flowing there is the potassium current. And it's this potassium current which is causing this hyperpolarization. Okay? So now, I better save a session. Well, let me do the init and run again and do a, no, I don't want to do a keep lines, just a init. And I will reduce this to 0.1 because that was the stimulus amplitude I was using. Do init and run. And let's do keep lines. And let's make that trace be blue. Okay. Oh, there we are. Okay, now init. We'll do a file save session just in case anything happens. Okay, now how do we rescue the spike? Well, we have to make the cell express some kind of sodium channels. And what ones are handy? These. Let's increase the density of those to 0.12 Siemens per square centimeter. Exactly the same density as we had for the native Hodgkin-Huxley. Okay. And now let's run a simulation. See what it looks like. Let's make sure that keep lines is not on. Okay, well, I can't tell what's going on here, except the black line turned blue. Init. Oh, so I am generating a spike. But it would sure look a lot easier to know. It should be easier for me to look and see what's going on here if I use color brush to change this color of the trace now to red and somewhat thinner. Okay. In fact, I'll make it even a little bit thinner yet. Color brush, red, even a bit thinner. Okay, so you can see that there's a red trace on top of the old previous spike that we got. The black trace is the spike we got with the native Hodgkin-Huxley sodium current, and the red trace is the trace we got by replacing that with the MyNA channel builder specified current. And if we look here in our plotted membrane currents, I'll do view equals plot. There we are. Let me do crosshair. Let me do set view. Oh, the heck with set view. Just let it be like it is. Looks good. Okay, are there any questions? So it looks to me like we've rescued the sodium spike. So the thing that we specified with our channel builder, first of all, we could use graphical tools that at a glance showed us the forms of the equations we were using and the values of the parameters that were there for those equations and the exponents that are in the equations that are used to calculate the conductances. And we didn't have to worry about any weird end model syntax. We didn't have to compile any end model files. And we've got something that we can reuse just by using h load file and then the name of this session file makes this thing available to any model, to any mechanism, to any model we want. And we can have as many different channel builders managing as many different types of voltage and or ligand gated currents that we like. And in fact, they can be stochastic if we like, okay? If we implement them as point processes. So we don't have to deal with end model. We don't have to decide, do we put the end model block first, or do we put the breakpoint block last, or where do we, how do we indent anything? Do we, are units working properly? Do we have units inconsistence? Don't have to worry about that. You can't make a mistake with the GUI. It won't let you. Okay. Okay. So, does anybody have any questions about the look of the appearance of this sodium current? First of all, did everybody expect to see that the sodium current and the potassium current have almost identical waveforms? I see a hand. Go ahead, speak. Tell me something.
No. I was raising my hands some time ago. Just wanted to ask, can we get the end model, like mode file from this GUI buildup? Like, can we export it into a mode file?
No, you can't, but why would you? I mean, it's just a block of, it's just a bunch of text.
Like,
It's a session file. You don't really need it. You don't need the end model code because it's usable as it
because if
is.
we have so many channels, and maybe once you build it and then later we don't need the GUI part inside, we can just close it and go.
Well, what's your concern about the GUI part? It doesn't impose a computational load.
It's an extra window I just want to get rid of, that kind of thing.
Okay, well, let's get rid of the window.
Oh, that's okay. It doesn't matter. Okay.
Okay,
I thought
it's there.
if you close it, that channel will not.
It still works.
Okay.
It still works. It's hidden. You can bring it back like that.
I see.
And we can hide it. And in fact, let me see if we can save a session file. Well, let's do this. Make sure it's in it. Okay, let me do a file, save session. I will call it hidden rig. Okay.
Mm-hmm.
And I'm going to expose Did I actually throw it away? Oh, I don't think I
think you had closed it by mistake instead of.
I think I'm right. I think you're right. Let me go back and let me restart. Yep. Python 3. Learn import. And I'll do a it'll be test rig. Sess. Okay, and now we do a init and run. And now if I, in the biophysics, I have 0 sodium density and 0 sodium channel density. So I can go back here and make that be 0.12. Okay, and I will init and run, and I get a spike, which is good. That's fine. So let me do this. Let me do file. Oh, I don't want to bust anything. I will hide this channel builder now, and I will do file, save session, and I'll write over the hidden rig sess file. Go ahead. Now, if I look in here, you'll notice that down here there's not a check. There's this channel builder is mentioned as one of the windows, but there's not a checkbox next to it, which means it's hidden, and I can reveal it if I want. Okay, and I can hide it. But watch what happens. Let me just quit, and let me try starting again. Python 3 from neuron import HGUI, and now I'm going to retrieve this time hidden rig. Hidden. rig. Okay.
Okay.
Oh, look at that. It is not hidden. Oh well, I can still hide it.
No, one more thing. One more
Now,
time.
here's the thing. Suppose you're running on a machine that doesn't have a GUI. Suppose you're running on a machine which is used only for computation, say like batch computation, like a high performance machine with many, many cores, and you don't need the GUI, and it doesn't have GUI. Well, Neuron doesn't care. These things, these session files, if they try to recreate a graphical element, and you're running on a machine that doesn't have a GUI installed in its operating system, and it can't do a GUI, it doesn't matter because it'll just ignore that part and it'll do the right thing with the computational engine.
Oh, okay.
Okay, the GUI part is only a user interface for people, and you can still have a command line which you can then use to initialize and launch simulations, even if you don't have a graphical interface.
But in case you have graphical interface, can we just turn it off?
No.
We cannot.
No.
One more time, I would like to have this in model export is that when you have a complex equation that is not in the preset equations, but you don't have to write from the scratch. Can we use the GUI to build a that mod file template thing, and then we can just change the equation part and make it a different?
Well, no, that's not yet available. Although if somebody wants to develop that, would be delighted to have it. I wish there was a tool that would produce well-structured end model code.
Just like the whole export.
Yeah, just like the cell builder can export nicely done hook for a either for a top-level model or for a class definition for a cell that you might use in a network. It would be delightful if there was such a tool that would do that with end model. There isn't anything like that yet. But I suppose if somebody has the interest and the skill, certainly wouldn't mind having it. Anyway, so the point is that you don't have to even use end model to add new ion channels. What you can't do, unfortunately, with the channel builder is you see when you use the channel builder's user interface and you're selecting equation types.
Yes.
You only get equation forms that make sense from a physical realizability perspective. And there are, if you go through the literature, you will find people who simply picked an equation and then bashed it with a hammer to make it fit their experimental data without regard to whether there's actually a physical process that might lead to something that's described in that way. And that kind of model you can't implement with the channel builder. You'd have to implement that with end model, because like a general programming language, end model allows you to do all kinds of crazy stuff, whether it makes sense or not.
That's
yeah, and even if it's just weird stuff that does make sense and isn't totally crazy, unfortunately, you can't do the totally weird things. So anyway, so when people say I'm interested in such and such a channel, and I don't see anything like it right offhand, I always point them, first of all, go to what's built in with Neuron, or go to end model, not end model, ModelDB, see if you can find something developed by people who really know what they're doing. And you can usually tell decent coding style probably means also that they know something about what they're doing as well, although clean code can be at its core not right either. So you try to make a selection and you try to make sure that it makes sense. And in extremis, we can look at something for you if you like. If you have a question, ask it on the forum or something. It's like is this an unreliable thing, or is it safe to use? Well, it's not going to format your hard drive probably,
Yeah.
unless they have a verbatim block that does something weird, and which verbatim allows you to embed C or C++ arbitrary code into your end model files. But I think in the future, I don't think Neuron is. It may turn out that Neuron is not going to be using verbatim anymore at all. So we'll see what happens with that. But if you find something that's close to what you want and you can modify it and make it work for you, that's fine. If not, then maybe it has to be done with by some other method. So.
No
Okay,
problem.
and so you saw an idea of an example of how I just worked through this and replaced the one with the other and showed that they're mathematically equivalent, computationally equivalent. Any other questions?
Thank you.
Okay. Well, with that, I think I will bid you adieu. And on Tuesday next week, we will meet again for the question and answer session.
Sorry.
I beg your pardon?
You wanted to discuss that NA current of the action potential.
Discuss which current of the action potential?
That sodium.
Oh, that peculiar current. Yeah. This.
Yeah.
Yeah. What about that?
I also couldn't explain the first, but in my lab we had a discussion and then found out that it's a reversal potential thing.
Okay. Yes. It's a good question to ask of a if you have a class of undergraduates who are reasonably interactive, or graduate students, or a postdoc seminar, it's a good thing to just bring out a whiteboard or a chalkboard or something and have people say, everybody knows what a spike looks like, and ask somebody to go up, volunteer to draw a spike, time course of a spike, action potential. And it's probably easy to get somebody to volunteer to do that. And then you have them volunteer, and you ask, okay, now can somebody draw a cartoon of what they think the time course of the sodium conductance is and of the potassium conductance? And you have them come up to the board, and then they draw that. And then you ask them to draw what they think is the time course of the resulting currents. Okay, so let's do the conductances here. Plot what? I wonder if I can draw. I wonder if that's available here. Sodium, GK, GNA. GNA HH. Good. And then we can do plot what? The GK, HH, and we do a knit and run. And of course, I know what the GNA is going to be here, because it's going to be 0. So let me go back to the biophysics and reinstate 0.12, and get rid of this 0. Okay, and now let's do init and run. Okay, so let me do a color brush, red for GNA, black for GK. Okay, and you'd be surprised what people think about the time courses of things. And if you compare the time courses, the question is, there's a lot of overlap here. And sometimes you'll see people will draw a brief sodium and then a brief potassium current, or conductance, I should say. And they'll even do the same for the currents. And we see here that they're very broad. Not only that, but if you compare them to the time course of the spike, Most of the current flow, let me see if I'm going to set this, do this right. Set view, okay. Set view 0 to 15, okay. Set view 0 to 15. Okay. Most of the current flow is after the peak of the spike. Okay. If I were to use this as a crude ruler, there's the peak of the spike, and very little of the actual current flow that enters very little of the actual current that flows through the membrane flows through the cell before the spike. Most of the current flow of sodium and potassium is after the spike, and the sodium current is very close in amplitude to the potassium current during that repolarization phase. That's very surprising, and people will get this wrong all the time. And then when they see this notch, if you're careful not to have the spike anywhere nearby where they can look at this, they'll look at the notch and they say, was that due to some, is there some other sodium channels available, some early fast channels available? Or is that some alternate gating scheme going on? Maybe these channels are clustering together and somehow altering their channel dynamics. I mean, these are suggestions I've actually elicited from a room full of people. And they're good ideas, but they're easy to verify. And if you simply plotted the driving force exactly as you suggested, you would see that the driving force collapses when this notch appears. Because we could actually plot, we can plot in this graph ENA minus V of 0.5. And that is the driving force for sodium current. And if I do that, and we do a view equals plot, you'll see that the driving force for sodium is pretty big, except right at the time around when that notch appears, when it is an order of magnitude smaller. It's about 11 down here. It's about 100 under normal circumstances, 111. And so that's why you get this notch. And the notch occurred at what time? It was right around the middle of the notch is right there at 2.8 milliseconds. And this minimum here is at 2.8 milliseconds. Okay. And so it's that collapse of the driving force that produces this notch. Not some alternate gating, not some weirdness in the equations being solved. Okay. But it's an interesting intellectual exercise to just do successive reveals of things and get people to draw things on the board and make their own predictions and then see how their own predictions are violated. We deliberately didn't do that today, but it's kind of fun and it's very illuminating. And it can be a dirty trick to pull on somebody during their oral qualifying or exam, or worse, during their defense of their dissertation. Not that anybody I know would actually do that sort of thing. Robert, you won't do that to your students, will you? I see a hand again.
No, I just, it was nice to see that we can write equations on the graph, like ENA minus V of 0.5.
Yes.
We can do any expressions there in the...
It depends. It depends. Try it and see if it works. Okay. That's all I can say.
Nice, I didn't know that.
Yeah. If generally speaking, things that you can get the names of in either Hoke or Python, you can usually get plotted in these graphs, but you got to use the You got to use the Hoke name for it, unfortunately.
Okay.
So it means you have to know a little bit about Hoke, or, hey, you know, nobody says you absolutely have to use neurons use a built-in interface. You could do this same thing with matplotlib.
Oh, is it? I didn't know. Okay.
Well, if you're running under Python,
You have to write the code inside the plot function inside. Yeah, I can.
yeah, sure, because on Python, what's the name of the membrane potential? Is S.V of 0.5. No? Oops,
Yeah.
I'm sorry. What is the name of my cell? Do I have a cell?
So
Let's see. Topology, H. Topology, it's called Soma. So it's Soma dot V of 0.5. Huh. No, it's not. It's H Soma. There we are. What? Whoa. Oh. Sorry. H0 Soma 0.5 V.
Yeah.
There we are. We need the name of the segment. V does not belong to Soma. V is not the thing that we're plotting. We're plotting V that belongs to the Soma segment. That's the name of the segment. So there we have it. So we could plot this. This is accessible to Python. We could plot this versus time. And then we could also plot H ENA. Is that right? No. H Soma 0.5 ENA. There we go. That's the name of ENA. So we could plot the difference of those two things. Right?
So first we have to record it and then plot it, right?
Yeah, you could probably plot that with matplotlib.
Okay.